Welcome back, everybody. So we're on to our last section for day one of the summit, and this is the roundtable discussion between the speakers that we had previously throughout the day. So please, again, get your questions in on the Q&A tab, and we're going to be bringing the speakers back to answer questions. So this is your chance to get any questions or answered or in front of the speakers. So maybe I can welcome them back. So Rathna, Marcus, Xiaoping, and Kevin, thank you and welcome again. Tony has gone to bed to get some well-deserved shut-eye given the time difference. So we're in good hands with the four of you folks. All right, yeah, and as the bottom of the screen says, please post all questions in the Q&A tab. So while we wait for some of the questions to come in, I'll open with one that's kind of near to and dear to my heart. So you folks are all obviously early adopters of technology, right? From your presentations that you gave and your overviews. So the question I have is, how do you bring your teams on board with new tech evaluations, recognizing it's a cost to you, it's a cost to the business? How do you go through that kind of sell job with your teams and that kind of the convincing, right? I don't know if someone wants to take that to start.
I can jump in, Jamie, because I'm old and retired and I can say whatever I want now. But I think it's not a top-down based approach, right? It's if you want people to buy in, they have to be part of identifying the solution, right? You just can't throw something to them and say, hey, now you're going to use this, right? And so you have to start, like the people that work at the bench know the day-to-day problems better than anyone else. And showing up with some interesting new kit and saying, hey, this is going to solve your problem may not really endear you to them at all, right? And so you learn a lot by spending time talking to people that do the day-to-day work, because they're the ones that have the problems that need to be solved. And they may not be aware of what the technology is that's out there. So I think that's the job of the leader, is to sort of be that translator between what the problem is in the lab and what the technology might be out there that exists. Because nothing goes better when people are totally sold and want to see it work, right? Versus being told, make this work, right? And so that initial identifying what the problem you're trying to solve versus here's some technology that may or may not solve a problem, I think is the first step.
It's kind of back to the definition of invention versus innovation. It has to solve a relevant problem or a pain point. Sorry, go ahead, Rathna.
No, I just wanted to tag along with Kevin, right? I think I'm in a space where I feel like I have some more advantage to what Kevin was talking about. Is I lead a method development group. So what happens there is they are looking forward to new and notables and trying to figure out what else can they do with respect to speeding up the process of developing methods or making their life easy. We work hard, but also we advocate for working smart. So in these cases, when there are leverages that we can add to, or showcasing things where a software, you don't have to wait for 3 minutes for it to load, where you process it and then you have your data to put together, because these guys are putting together updates every week for our pharma biotech clients. So they really value that, to see that they can save time a lot of the times, and then they can be exposed to a lot of different things out there that are new. And I think they are very passionate about talking about it to their production colleagues and saying, oh, we tested this out and this looked good. So I think it gives them a lot of satisfaction in their job when they do that. In that sense, I feel like I'm specifically in that position that I feel very lucky to be in with my team as well. Like listening to and adapting to these new technologies ahead of time as well.
Well said. Well said. Thank you. Thank you both for your insights. I don't know if Xiaoping or Marcus, you want to?
Yeah, I can add to that. I think in the ideal case, people go to a seminar, they read the publication, and then my collaborators come and say, hey, I saw that, we need that, right? And then we implement it. So that's the best thing. The other thing is a little bit tricky, right? If you come with, let's say you come with the SCIEX OS, right? And then you say, okay, top-down decision, we switch now to SCIEX OS, right? Then you might have the team which tries to prove you're wrong with this decision, right? And that's exactly what Kevin said, right? It needs to come from them, right? Basically saying, hey guys, there's SCIEX OS, look at it, decide on yourself if this new function really works for you. And once they decide, once you lead the horse to the water and they start training, then we can go for it. That's basically the process. But top down is tricky.
Yeah, seldom ever works, I think. Yeah. Maybe in that vein then, so that's all about getting the team's buy-in or followership for the new technology. So in addition to that followership for the new technology or the tech evaluation, what other considerations do you folks think about before deploying innovative technologies into your existing workflows or SOPs?
Yeah, maybe I can start on that too. I feel like it's important to understand if this process is something that can be a add-on to what you're doing with respect to, is it a sequential process for you that helps you with your everyday work? Is this something that you as your team or as the company is looking ahead to what it wants to bring in as new and notable technologies to improve upon the offers that we have? I'm speaking here from a CRO perspective. So we look at advancement in technologies or where the market is today, and we wanted to bring in these new technologies to adapt to offering these new things that we can do to answer some of these questions that maybe our pharma and biotech colleagues have. Those are the things that will excite us to go ahead and put in the time and the work to try them out, to be able to see if that is feasible for us, as well as that way we are exposed directly as well to understand, is it something we can easily incorporate to our current working processes with minimal sense of change, to then get to a place where we become the experts in the field.
Makes sense.
I mean, if you look at, for example, at the Echo TOF system, right? Why would somebody invest in that? First of all, you need to have the need for this capacity, right? Because otherwise you go and say, hey, guys, I have now this system, I can do 1 million compounds a day. And then like, nah, we don't produce 1 million compounds a day. So it's the whole package, right? We say, oh, well, we need less protein, for example. This is an argument which is very strict, right? So you bring something new to the table, which is really giving some, but you can do assays. Let's say, for example, in my example, we could never do like a full assay because it was taking too long. But if you now go in and come with an instrument and say, now I can do 20,000 compounds easily with mass spectrometry because that doesn't cost me nothing, basically. Then this is new capability and this makes works investing in it, right? So it's really always looking into it. We and probably the CRO is the same. It's fit for purpose, right? If you come with an instrument which has this super performance parameter, we might just say, yeah, but we don't need this, right? We don't need this highest resolution, but just fit for purpose, right? So it needs to give additional capability, which allows us to make decisions which we couldn't do any other way. That would be the case for us to jump onto it.
Yeah. I think part of it is people get so used to doing things a specific and certain way that when a new technology is there, they think about how do they use it to do the work they've always done, and don't think about different approaches. Like the Echo MS is a good example. It's like, well, we compromise how we do experiments because an LCMS approach doesn't allow us to have the throughput that we need, say, like, I don't know, a time-dependent inhibition assay where you could generate tons and tons of samples, but if you had to do LCMS, you couldn't do it. It's not practical. But an Echo MS, so it's about thinking about, not about how do I do my same experiment, the same way with a new technology. It's about how do I do a better experiment with new technology? Because all the experiments we do now are compromises, right, on how we do them, right? And so, that's, I think, the tricky part is getting people to, step one, fire all the people that said we've always done it this way, right? And encouraging people to think about doing their experiments differently, right? And that gets to the SOP. Who wrote the SOP? Well, you did. So you could change your SOP, right? Don't use it as a crutch or an excuse not to change, right? And so I think it's the trust of your scientists to let them think about doing experiments differently based on the technology at hand.
The trust and the freedom not to be constrained by the past, I guess, right?
Yes.
Where you come from. Yeah.
I think the implementation is really based on the initial need. As I say, when we bring new technology in, they either could be there's a gap you have to fill in, or a lot of times it's just improvement, making better, more efficient. But depends on the need. A lot of technology is brought in. It's important. It already pointed to how we implement. But in order for something to improve, you have to have people see the same thing. So if it's some, the product development required is demanding, you need a new technology platform to fill in to accomplish the task. Then that's a lot of like in some of the collaborations we did. We have this ICIEF method across the industry used as a release testing, but we have no way directly to assign ID. We need a new technology as a collaboration of the instrumentation company, university, and the pharmaceutical company together. Develop this message to now we can bridge the gap. But a lot of times it's really just getting better, improve, or if it's a new whole technology development, then it improve hardware, software, and all together. I think the implementation is really tied to the need. As the technology, the data speaks, right? If this whole data package really produce proof is better, is more efficient, it save time, energy, money, then that's just have to like platform like this to share.
Yeah.
It's a win-win situation.
Yeah. Thank you. Let me see here if we have some more questions. There's one here. I might have to reword it a little bit, but it starts as what non-MS based innovations might be or should be on the horizon to really impact quantitative workflows. So source technology, software innovations, sustainability improvements. So I would say maybe just in general, what MS-based innovations are you looking for? Back to the problem of valuable problems, right? What are the pain points that you see, and what dimension in a mass spec would you like to see improved upon to address some of those pain points? Kind of a wide open question there.
It is, right? It always strikes me like we go with this miniature amount of samples, and we have these huge machines, right, which go and which we utilize to measure them. So it's not elegant, isn't it, right? So basically, why can't we have mass specs which are the size of a rice corn, right? And then basically we directly put them in the animals, the real-time sampling, something like that, right? So it's like we go like sup, femto, atto, iotto, septomol, right? And still have these huge machines, right? So you think like this is a big overkill. And then you have like 1 million machine to look in one single sample, right? This is also something which is like, it can't be, right? It should be different, right? We should be able to have them in parallel. They should be much cheaper, so we can do much more with smaller instruments for less money. So that's basically the big picture I see. Right?
The size of your instrument is inverse to the LLOQ at the end of the day, right? So
yeah, it's huge machines.
One thing I thought about is to this non-mass spec-based innovation method to improve quantitative workflow. I think automation, mass spec based, but automation when utilized, correct, it's really improve the quantitative and also manpower.
So, and when you say automation, is it just broadly speaking? Is it sample prep? Is it the data processing? Is it across the board? But if you had to pick one, what's your pain point right now for automation?
Very good point. We could use for sample prep. We could use the existing or ever developing robot. Taken Hamilton sample prep or throughout. The dedicated sample TDS preparation. You also mentioned, I would say cover both. For example, the software, especially come to mass spec software. Me come from a non-mass spec background. I would really appreciate mass spec software can be straightforward or simpler than it is. I appreciate the immensity and the capability of mass spec software, either Biological Explorer or Gene Database or other, but it is very complicated. If we can, I would not necessarily use the word automation, but streamline it, that would be, in that category.
So I think sorry.
Go ahead, Marcus. Go ahead.
Oh, yeah, you.
No, I was just thinking, in order to add to what Zhu Ping is saying, what I was thinking is AI in integrations and performing reports and things like that, that area under the curves, the way we draw it, maybe it can learn from the earlier part of feasibility work that we do, and it can start integrating it for us. So we just review rather than we integrate and we review, right? So same thing with respect to statistics. Instead of going through pulling in the batches to regress and to be able to then define the curves and the QCs, for example, specifically towards quantitative mass spec, the AI can learn that while we start working on a project and adopt to it. So there is less work involved and it's much more automated, is what came to my mind when we were talking about it.
Yeah, I just wanted to add more. So basically, solutions is the thing, and I think SCIEX and other companies are going this direction, right? Instead of just selling an instrument, you do whatever you want with it. We all have the same kind of similar issues, right? We want to, do protein profiling. We want to do atomy. We want to do PK assays, and so basically, if with or lipidomics assay or whatever, if you come with a solution which runs out of the box, that's basically the thing to go, right? So instead of having, okay, here's a piece of software and then you stitch it together, and that also requires that people try to rethink, right? Currently, we still have people who think, well, I need this software to do that and that. But basically, if I could, for one workflow, buy the full package from a company, that would be a big, big cost saving for me.
Thank you, folks, for your insights. Here's a fairly technical question for one of you. What typical LLQ for a hybrid LCMS on protein biomarkers, PD makers? And is there a relationship between the LLQ and the protein size for quantitative LC MRM-MS methods?
Yeah, I can take that question, at least start to take on that question, I guess. So with respect to relationship between size and LLQ, not really. I don't think there is a direct or indirect relationship to that. With respect to the LLQ that we typically go after, it's multiple fold. It's not as straightforward as figuring out, oh, you need a high nanogram per ml or a low nanogram per ml LLQ. It depends on what you're going after. For example, in the case study I presented today, we are going after a total peptide quantitation or indicator to a total protein, as well as a human specific marker, because it's a replacement therapy product that we're going after. So maybe one of the peptides we are looking at maybe doesn't need a very low LLOQ, but the other peptide as a part of the same assay may need a super low LLOQ because it is not present endogenously versus the other part of the molecule is present. So maybe a 10 nanogram per ml LLOQ is good enough. But how do you define it is twofold. One is you look at the literature and see if there are any profiling that was done even by an LBA assay to see generally what are the levels. So based on that, you define that if you don't have any other data. If that is not the case, you go to the best LLOQ that you can get with an assay and then run the samples to see if that is, how much percentage of your unknown samples fit into this range of what you're working with. So it depends on how much information is available versus not. The other aspect that you want to consider is the type of matrix you're working with. For example, you're working with something that is supposedly working in the brain, so it has much more availability in a brain region for a tissue. Whereas in plasma, maybe there are some circulating levels, but the amount that is present is super low compared to a brain tissue. So those are the things you want to take into account depending on what the ask is. If it is a biodistribution versus a PK study, things like that. I think those all make a lot of play a big role into figuring out what that LLOQ looks like.
Thank you, Rath. One, I guess, directed towards specifically Kevin and something you said during your talk there. So you mentioned that we're the author of our own SOPs. However, this particular person has been getting pushback from regulators in terms of new innovations. And how do you balance regulations versus innovations and kind of changing those SOPs at the end of the day or recreating them? What's your experience there?
Well, I guess it depends on the technology that you're trying to implement, right? If you're working in a regulated environment and you're using a technology that doesn't meet the criteria, maybe the software is not 21 CFR Part 11 compliant, you're going to get pushback, right? But if the technology it meets the criteria, then it's the regulators, in my experience, are open-minded and willing to have that discussion around the technology, right? If we look like dried blood spots, for example, is a hot button has been, but when I was at Merck, we took several programs to regulators, presented the data, and they're like, yeah, looks good, right? And so I think sometimes we use that as an excuse not to do new things. But I'll caveat that with, if it's a technology that doesn't meet the criteria that regulators lay out, you're going to get a problem, right? And mostly it's related to software and data integrity, right? But a new mass spectrometer that's never been used isn't that new, honestly, because there's not a lot of new in mass spectrometers from what is already out there. But if it's, say it's SCIEX OS and SCIEX OS didn't have 21 CFR Part 11, you're using analysts, then of course you're going to have a problem because you can't show the data integrity. But SCIEX OS is, so it's fine. But if you're using some small company that invented a new a new sample prep technology that requires and generates data that you require to show the integrity, then you're gonna have a problem, right?
I saw a lot of nods there from the panelists while you were giving your answer. So Rathna, Marcus, Xiaoping, is that your experience as well?
I echo Kevin to the core. Like I echo with him, like every single word he said. A lot of times I think people who have been doing it for a long time, most of the time I find that want to keep up with the same things that are written in the SOPs. I think what Kevin was getting at is you wrote the SOPs, meaning you read the guidance and you wrote an SOPs based on that guidance, right? And that guidance can be interpreted multiple different ways, to be honest with you, depending on, for example, they ask you to I'll give one quick example and I'll pass on to the next panelist, is when you have a curve fit that you're working with, it calls for simplest best fit. And I've had a lot of team members in my past ask me, I always only want to use linear 1/x square and don't want to use anything else. And you do that because linear 1 over x square is the simplest best fit. But when it doesn't work, you figure out what the simplest best fit is. And if that's quadratic 1 over x square, it's okay to use quadratic 1 over x square. But I think to understand that fact, it takes a lot, right, sometimes to get through. So I definitely echo what Kevin was saying. Thanks.
There's always a resistance to start anything new. Again, you know, if the technology, if the package, the summary, the data is solid, it's a matter of communication. In the pharmaceutical world, I think it's always starting from FDA, you know, to any new technology. You have to prove, right? You better, why you better. But once that is, you know, established, in terms of the SOP, this test method, SOP, I think each company just have to have that, you know, regulation established. And in the initial stage, it always take a lot more effort and time. Yes, not way, you know, if I'm the one to start to evaluate some technology, I have the opportunity to be there for a long time, so I can see the advantage. But that advantage, not, you know, the better part, improvement have to convey to including regulatory. So once they see that, then there is a reason, a solid reason, and that's where the SOP is established.
And I'm in a very comfortable situation that I don't work with SOPs, because I'm in early research. We basically don't care. We just want to get results, right? And if SCIEX has a new instrument and whatever, we just try to get results out of it, we validate it, and then if this result is good, we present it to the project team, and that's it, right? And that's the beauty about it. So basically, we don't have any SOPs which are directed, but it's good scientific practice, which we obviously need to adhere to, right? So basically that we come to a decision board meeting with data which is sound, even if it's a prototype instrument, right? So that's driven by just our scientific education.
Same process, data driven. Thank you, folks. There's a few minutes remaining. Maybe I'll second last or last question. It'll be a non-technical one. So which academic degrees are most attractive for your groups for hiring? And also, what do you think about the on-the-job training concept?
Yeah, so I don't think there's a specific degree, or, you know, like in terms of what you're looking for. It doesn't, to me, there's a job description that's written. If you're hiring someone to do small molecule LC-MS analysis, then you don't need a PhD in, you know, from a big, you know, lab where you learn mass spectrometry necessarily, right? I'd rather, you know, see someone that has a bit of experience, maybe over their undergrad in different, you know, different labs, like summer interns, because there's lots of opportunity for those types of jobs where you can apply and get a summer job, right? And so that, I think, is more valuable than what degree you have, whether it's a biology degree, a chemistry degree, you know, a math degree. It's really having a bit of experience on your CV and apply for those summer internships. And I know if people, you know, if there's people looking, it's now that you apply for next summer. And people may not be aware that they might, hey, I need to do my job search in January. No, like at least at Merck, those jobs are posted now, and you need to go apply now if you want a summer intern at Merck next summer. I don't know what other companies are like. And then beyond like undergrad and after maybe a PhD, there's postdoc programs as well, which then industrial postdoc give you sort of a sneak peek inside pharma, and it's sort of an opportunity for pharma to assess you and you to assess pharmas, because a lot of those postdocs get turned over into permanent positions, right? And so what I'm saying is, it's not a specific degree, it's the experience that you get throughout your degree that makes you an attractive candidate.
And the interest in this technology, right? And so in Switzerland, we have a lot of people who do apprenticeship, right? And so it's a 3-year apprenticeship, and in there they learn how to work scientifically in the lab, and they develop, if they have a desire to work with instrumentation, right? That's a big deal in mass spectrometry, right? You need to be able to work with instrument, get a feeling for it, work with data, having this understanding. And so in our business, we don't really pay too much attention if it's a apprenticeship, a bachelor, or a master on the basic level. Obviously, if it comes down to project work, then it's different. But just working in mass specs, we have people on all levels, juniors, seniors, whoever has fun, and it's interesting. We have some people who want to spend their life with mass specs. You cannot promote them, right? And avoid this. We have this big deal that we always need to develop people, and some people just don't want. I want to stay in the lab. I want to work with my mass specs, and that's where I belong. It's also an interesting aspect.
Thank you.
Yeah. Oh, I just wanted to add to that is I think if, again, degree is not of essence. I think if I were to pick the major and be very specific, I'm looking for somebody with a chemistry background with a chemistry major with a mathematics or a computer science minor. I think that fits very well with what I want in my team, generally speaking. I know that those are very specific, but just wanted to say that, as well as with respect to on-the-job learning, all the time. That is kind of expected. I would think that 85% of what we do every day on the job learning. So.
Yeah, I think I saw a lot of firm nods, including my own, with that one. So I think with that, I'll wind up the panelist discussion for today. Thank you, everybody. Thank you for your time and your participation. So really appreciate it.